Thank you and welcome everyone. It's my pleasure to start off part 2 of the Rigor and Reproducibility series. This series started back in February when the Office of Sponsored Programs, Laura Johnston, and the Office of Regulatory Affairs recognized that rigor and reproducibility was going to be a topic that is here to stay and going to be affecting our grant submissions and grant receptions as well. So we thought it'd be a good idea to start the conversation on campus and try to raise awareness as much as possible. And I think it's taken off in spite of us. So that's a good thing, because that just means that the science from Michigan State University will be getting better and better and better. Today, we are splitting this presentation into 2 parts. We'll have a short period about use of animals and as rigor and reproducibility applies to animal-based research in a general way. And then we have the pleasure and honor of having Dr. Cindy Jordan and Dr. Marc Breedlove, who are specialists in sex as a biological variable, to talk about that topic and how it relates to the whole granting process and scientific process. We are looking forward as well past today and hope to bring other topics to you, so stay tuned. So let me get started. As I said, we're splitting this up into 2 different presentations, and Dr. Hankenson and I will do the first one. So, and I'm just going to give a general overview in terms of what's going on out there and some of the resources that are available. So this slide simply shows a lot of the publications that have occurred over the last 4 or 5 years on this topic. This has just been accumulated. In fact, if you go back and look at the journal Nature, there's been an article close to every 3 or 4 weeks on this topic. Not just animals, but rigor and reproducibility in general. And they've been rather creative in some of the titles. They do capture your attention. I particularly like this one, why most published research findings are false. So it's a good article, but a little inflammatory. So moving on, the general review criteria that NIH has kind of laid down for all of us to consider in terms of rigor and reproducibility include the importance of the scientific premise. How good is the basic argument that you're building your research program off of? Rigorous experimental design. This includes both design and statistical analysis. And this is a topic that we could go for days on. Consideration of relevant biological variables, and this also includes sex as a biological variable, that both of which we'll be talking about today. And then the final area that they have drawn to our attention is authentication of key biological and/or chemical resources. This would be cell lines, transgenic animals, wide variety of topics here. So these are the general criteria. As I said, Dr. Hankenson, Dr. Jordan, Dr. Breedlove will go further in the topic of consideration of relevant biological variables today. The only other thing I wanted to point out is that there are lots of resources out there for you to take a look at. And these slides are going to be posted on the Vice President's website, so you don't have to scramble and write everything down here. There have been a number of recent publications and comments in major journals. Professional society recommendations, FASEB, American Society of Cell Biology, Society for Neuroscience, and many, many others. There have been a number of general guidelines and standards that have been put out. These are both the ARRIVE guidelines from Europe and guidance from the National Academy of Sciences in this country. And then a number of workshops on the topic, and these workshops have resulted in some really good resources on the web. Both ILAR, the Institute of Laboratory Animal Research at the National Academies, and the American Physiological Society are two such websites with good resources. So with that, I'm going to turn the podium over to Dr. Hankenson. Let her pick up from here.
All right, so I'm curious how many people came to the February session. Was anyone here? I know Laura was here. Yeah, good, a few people. Okay. Well, welcome back. And for those of you who are coming today for the first time, I'm going to review some of the things that have come up in the literature since that time. A couple of interesting articles that we'll make available to you after today's session. And then also I'd like to generate a little bit of discussion as well. So one thing I do want to ask, how many people work with rodents, mice or rats? How many people work with non-rodents? Okay. How many people came just for the snacks? There's a few of you, I know. All right, Greg, it's okay. And so what I feel, and JR and I were just on a conference call yesterday with the FASB working group, and while a lot of this information is really focused on rodent studies and the reproducibility in those species, largely because the majority of the work that is being done and through the NIH is in those species. I also wanted to make sure we're keeping in mind those of you who don't work with rodents, and I'd like to hear more about the issues you may be having, because this really should be applied across the board when we're talking about reproducibility of studies. So with that, here is a standard rodent housing room. For those of you who have not seen this before, this would be a caretaker down at the bottom who's pulling out a mouse cage to look at the animals to assess for health. And I just want to throw this out here, the general question, what would be the potential contributors at this level that could contribute to irreproducibility? So just throw them out. Things you might think. What could be a variable based on how, all the same cages, all the same bedding, all the same food. Yes? Absolutely. So caging density is very important. And do you have any thoughts on why that's important? Yep. That's a big one. The temperature, yep. There's a lot of models that don't show up based on cold stress versus heat exposure. Yeah, definitely. Good. Yes? If they're all siblings. So inbred versus outbred, how related they are genetically, absolutely. The location of the cage. So that's a really interesting one. I'm so glad you said that. We get questioned when we have site visitors coming through to look at our animal care program about the placement based on the light exposure. And we've been asked before, are we rotating cages from the top that are closest to the ceiling lights down to the bottom? And if you start thinking about what it would take to move, that's Ola, think about what it would take to move cages enough to give them all the same exposure, that in and of itself is a huge variable because moving the cages has been shown to be quite stressful to the animals. Any other thoughts? The sex of the animals, absolutely, which will be coming later. Sex of the caretaker, yes. So the personnel is a huge issue. Pheromones that are given off have been shown, perfumes, deodorants, those can have effects. The olfactory studies that may be going on behaviorally, these are not things to take lightly. But we are not able to standardize on only the lab personnel or the animal husbandry personnel to always only have one particular individual. It's just impossible. Any other thoughts? Enrichment. So we are expected to give some sort of device for rodents. For mice, typically it's a nest building material. For rats, it's some sort of chewing toy, like a nylabone kind of a thing. So what kind of exposure they have to that. Again, how many animals are in the cage that get access to that? How about bigger picture outside of the cage? Yes, noise in the room is huge. Vibration in the room. Light cycle. For those of you who have rodents on campus, do you know the light cycle for your room? Should you know the light cycle for your room? This is really interesting thing about how deeply can we go on the analytical detail. So 12-12 is how we would describe it. 12 hours on, 12 hours of lights off. Temperature and humidity has a range we have to adhere to. And in middle of Michigan, we have hot months, we have very cold months, and that does impact how the HVAC is working in our buildings. Anything else with the room? Humidity is a big one. And the humidity in the room is going to be different than the humidity in the cage, depending on whether the cage is ventilated itself or a static box. So there's just so many things. I just wanted to use this as one small example of considerations that just are happening at the room level, completely outside of the experimental variables that you may be looking at. So this was an article that was sent out by my colleagues, my lab animal veterinarian colleagues, about never wasting a good crisis. As JR said, these have catchy titles. And what this article basically was talking to was what we all have been relaying. Where are the Achilles heels of the experiments that we're doing? And so in summary, there's a nice figure, and I'll just kind of give a summary of it. And this will be an article that I'll provide for everybody, and actually has nice hyperlinks to other articles and resources. But big issues are the reagents that we're using. So for example, are the antibodies vetted appropriately? Have the chemicals that we're given, are they pharmaceutical grade or chemical grade? How do we look at the methods that are being undertaken? How many of you have given your animal use form or protocol to the person that's doing the experiment so that they have some sort of recipe or model or something to follow? My gut feeling, and from how it was when I went to graduate school, was you just learned from the person that had been doing the experiments across the lab. You didn't necessarily get the instructions from the animal use form. And I hate to say, when I lecture to vet students and I ask them how many of them as summer students have gotten their protocols, it's very rare for a hand to go up. So we're not, I think, as mentors, necessarily giving the information that we need to folks. So then they kind of figure it out on their own. And often that practice is going to be a variable in and of itself. Within the cage itself, we talked on these a little bit, for rodents in particular, litter mate controls, what the genetic background is, and statistically, what tests we're putting on these studies, what kind of randomization, are folks blinded to the experimental variable or not? Those sometimes you can have a bias, and NIH has been talking more recently about the statistics that we do often after something has been studied may actually lead to a bias where we are confounding our own work and looking for what we want to see and publishing what we want to see. Other things that can happen just through virtue of doing research. I think I mentioned in February, my mentor told me it wasn't research until somebody was crying, because it's always so hard to repeat things. But contamination of tissue culture, you have trouble with infection that may be going on with your animals, the culture conditions, how many times they've been passaged, the media. Again, all of these things can impact outcomes. And very few times do we see that that level of detail is able to be reported. And we can talk about why that is as well. So this was a good article. I think that you would enjoy taking a look at. And like I said, I will make it available on the resource site. And then the other one that I wanted to touch on that also has come out in June of this year was just discussing, again, you'll see the same names on many of these papers. And John actually, I think, came and gave a lecture at MSU probably about 18 months ago now. It was quite a provocative talk. But what does research reproducibility mean? And again, these big ticket concepts that we are discussing at this point. I just want to give a little bit of a highlight to some work that a few of us, JR, myself, and Laura, were cited in and interviewed really by a science writer this summer. And it was really interesting talking to her because she herself had not thought about the idea of reproducibility and the fact that even just the microbiome of the animals, so the basic gut flora that our animals house, no matter what the species, can impact how they respond to infection, how they interact with the experimental variables, how well they gain weight, et cetera. Laura, I don't know, I might tap you for 2 seconds to give the quick soundbite about what you found, if you don't mind, with the variability that you had.
Sure. So one of my grad students did a study. So he
You want me to give you?
Sure. So one of my grad students did a study, and he got a result. So the thing that we were doing increased bone density. And so he was very, very excited. So he'd show me the data, and I said, well, let's just do this one more time. So he ordered the same mice from the same vendor and repeated exactly the same study, and he got bone loss. which, that's our output. So I'm like, whoa, this can't be. One of these has to be a fluke. So let's repeat this one more time, and that'll tell you which one is really the right experiment. So he went ahead and did it again, and he came back, and he was just like, oh, it was so sad. I'm like, what's wrong? And he said, there was no difference this time. So we got three different results from the same mice, from the same vendor, the same age. Everything was the same facility. And so we've been looking into using that, though, because it's very powerful, because actually we got three different results and everything else is the same. So now we can look at the factors that correlate with the different outcomes in those runs. And so so we were interviewed on this one because I think they found that really funny and interesting. And so we're working on that. And it turns out that we're tracking the microbiome and changes there. There seems to be a particular microbiome that affects the outcome, and we can transfer that to mice and get either increased bone density or not. And so that's sort of the direction we've been going.
Thank you. So the microbiome is becoming, I think it's been an area of study, but for laboratory animal science and people in my field, we hadn't really started to analyze this until about 18 months to 2 years ago. And Craig Franklin at University of Missouri, who is my counterpart there, is doing quite a bit of work to look at this. And essentially, when animals are coming from Jackson Laboratory, let's say, or from whatever vendor, within a matter of a week to a month, the microbiome has shifted completely because they're put on a different type of bedding often, and some of that they'll ingest. Their food is often different. They've switched from what they were having at their home site to what they get at their destination. And what we're finding as well, even though within our facilities, and we have at least 7 rodent facilities on campus, we have variability even between facilities on campuses, and not just at MSU. This is something that's across the country as well. So very much keeping things the same is a great option, but much more it's just defining the parameters that you have, because I don't believe we'll ever achieve standardization because we aren't standard. The people factor also comes into that as well. And so then overall, the fact I think that I feel is a big one is that our publications don't let us offer this information to one another. And from the first paper that I showed you, these are just some factors to think about. We have a tendency as an academic population to generalize the positive results. Often experiments are repeated because we're inherently biased to look for those particular results. And we don't necessarily report negative results enough, if at all. And so if you have other scientists that are trying to emulate things using just slightly different approaches, it's going to be very challenging to reproduce exactly those results. We don't have a journal of negative results, to my knowledge. We do? For how many disciplines?
Maybe
2 of them, okay. So maybe we have 2 of those. They're usually not the most popular journals, and I don't know if everyone's reading those, but that's the problem too. We're so limited by our editors on what we can put into manuscripts. Some of the proposals have been to actually allow for appendices that have this greater level of detail. And then, as I mentioned in the final point, we just are having trouble even getting observations at one facility on campus to be emulated at another. And again, teasing out those often requires a great amount of mice, a great amount of money, and money is very difficult to come by. So I just wanted people to think about that. JR had mentioned that the FASB group had done roundtables, and so some of their other suggestions, just to share with you, are to develop lists of variables to consider when you are setting up your designs, clear descriptions of what's important to report for your particular disciplinary field, the consideration of sex as the variable, which we'll hear more about, and overall, I think just better communication about how these studies are being done. And just as I conclude my part, what I'm most concerned about in my position and for my role nationally in lab animal medicine is that with the increased discussion around the inability to repeat results, there will be a perception that research is not valid, that what we do is not worthy, and that funding will be pulled away. So I'm hoping that we all can advocate for how that the science is important, that the variability is okay, as long as we're defining those parameters around which they happen. So I will stop there until we have discussion, and I'll pass it over to you. Who's next, Cindy? Good. You need that.
How's this? Is that good? Yeah, right in the middle. Thank you. Okay, so as Claire warned you, we're moving on to sex as a biological factor. I'm going to provide some of the snippets from, here I am moving away from that microphone, but let me lower this a little bit. I'm going to provide some snippets from this guide notice. Here it says, failure to account for sex as a biological variable may undermine the rigor, transparency, and generalizability of research findings. Okay, so this business that if we test only males and we develop drug treatments for only males, and then we find when they actually go past and receive FDA approval that they don't always work in women, right? And sometimes they're actually toxic. I mean, it's not only failure of efficacy, but in some cases they're actually dangerous and they have to be pulled off. And so it is a real concern. So here's a little bit more from this guide. This was released in June 9th, 2015. And NIH has been talking about this for a while, but it has finally become real. And they're getting serious about it. And so the first thing, of course, is to release a policy. So NIH expects that sex as a biological variable will be factored in to research designs, analyses, and reporting in vertebrate, animal, and human studies. And we've seen that. As a sex researcher, I've seen an awful lot of research on males. And there are some non-truths around why only males have been studied, by and large, and Marc will be talking about that. Or the other thing, which I think is equally severe as a crime, is not to report sex at all. And so as a sex researcher, I know that, I deliberately am in seeking out things that have sex differences, but in fact, it's often very hard to study something that doesn't have a sex difference. Okay, so strong justification from the scientific literature, preliminary data, or other relevant considerations must be provided for applications proposing to study only one sex. And of course, some of the obvious examples is, I want to study prostate cancer, right? You can't study prostates in women. Okay, so at the bottom of this 2 to 3 page guide, there's a list of resources. And I think I've read basically all of them, and most of them are pretty good. But there's actually one that I really favor, and that's because it's part of the Journal of Neuroscience Toolkit, and it's giving you a roadmap of, oh my Lord, I found a sex difference, what do I do, right? So it's really helping you to figure out what are some of the fundamental questions that you need to attack to understand what's behind this sex difference. This is a paper that, in fact, it seems like a lot of the non-researchers, so it's the FASEB paper, a lot of the researchers that don't actually study sex are aware of. And I read this paper, and it takes a very interesting position. So the title suggests that it's going to help you to learn how to study sex differences or how to deal with sex as a biological variable. But in fact, it spends a lot of time arguing against or explaining to you how hard it is to do that kind of research. Okay, so what Marc and I have planned is that first I'm going to start with the FASEB paper, And we're going to talk about that. First steps for integrating sex and gender, right? So that sounds very promising, right? But the basic thing is, well, yeah, it's too complicated, right? And I can imagine that this is kind of where one starts if you're not used to this idea of thinking about males and females, right, and how to deal with these potential differences. This, on the other hand, is a clear message of it's not so complicated. You can really do this, right? Which is why part of their title is it's not such an inconvenient truth. Okay, so let's chat about the FASED paper. So they start by tackling this big issue, which I have dealt with as well. Do we say sex or do we say gender? They do have an opinion about this, and NIH happens to agree with it. So can gender be addressed at all in laboratory research? What they say is that sex refers to traits thought to be primarily based on biological characteristics. From the guidelines, NIH, defined by characteristics encoded in DNA. Okay. Gender, on the other hand, is inherently sociocultural. Psychological is what I say in parentheses, construct, right? So it's this idea. And it's geared around our social, they talk about gender identity, right? And then NIH clarifies that a little bit further and says, refers to social, cultural, psychological traits linked to human males and females, right? So if you're studying humans, yes, you better be thinking about both gender and sex. If you're studying animals, I think it's pretty safe to say that you're referring to sex and you're studying sex. Now, where gender has come into the conversation in regard to animal research is something that Claire referred to, which is in thinking about the gender of the animal caretaker, gender biases in terms of how we as scientists are dealing with the data. And it's like, I don't even want to go there, but I did a recent application on a relatively new field for me, and it's like, do I say gender, do I say sex, right? So if I'm confused about what to say, I figured maybe some other people are confused. So animal research, I think you can call it sex differences. Okay, so these are some of the reasons discussed in the Ritz paper of why it's too hard, or worse yet, raising the possibility that it's in fact irrelevant or distracting to include sex as a biological factor. And this is why I get confused about this paper, because I end up not being clear on what the true mission of the paper is. And so it's interesting to me that it's listed as a resource, and so this is more a warning more than anything else. Okay, so one of the things is it takes a lot more money, resources, know-how, right? So they're putting that out as an excuse, and I've certainly heard people say, we can't afford to do this because it involves so many more animals. And so if you're used to doing males and you have to do females, and then you're studying rodents and they have a 4-day ester cycle, and so now you're thinking about 4 days across the ester cycle, you're looking at 5 times as many animals if you want to achieve the same power, statistical power, to detect differences, right? So that's a lot more animals. And that is problematic. And one of the places where I'm still waiting to see what's going to happen is NIH actually prepared to support this mandate by resourcing scientists to do the kind of work, rigorous science, that they say they want us to do. Distraction, not really relevant to what I'm interested in. I'm studying disease. Why do I have to study differences, look in both male and female? Well, one possibility is you could find a difference. And if you find a difference, we have some ideas about what might be behind that that difference, and therefore you end up with another avenue or tool or way of understanding the disease process. And not only that, this is normal. It's biological, as opposed to some of the other manipulations we do, which is to, insult the system with some toxin to understand a disease process. Okay, immortalized cell lines may come from a tumor of a man or a woman, are likely to be different, independent of the sex of the donor. So immortalized cell lines, I think, offer a genuine challenge. Oftentimes, the sex chromosomes are lost. Another thing that happens with immortalized cell lines is, there's this one special tumor that has a particular phenotype that has been, the original donor is male, right? And you may, in both cases, be looking at liver cancer cell lines, but in fact, the details of the cell phenotype and genotype are different between the male and female. But I think more specifically, the details, I haven't said that very clear, is that liver cancer, the cancer itself can vary, right, when it comes to who the donor is, quite independent of sex. So that becomes difficult. And so my recommendation there is, of course, you use good grantsmanship and you acknowledge right off the bat, you don't try to hide this fact, that this is a drawback of your model, and you absolutely champion its strengths, the history of the model and what has been learned, and clearly lay out a plan for how to address this issue of sex in future experiments by considering going to primary culture or something like that, right? So you have to have a plan, is the message. This is a quote directly from the paper. Sex is not a fixed property of cells. I struggled a little bit with understanding what it is they actually meant, but I think it has to do with this fact of genes versus environment, right? So we know that the genes, the sex chromosomes, if we eliminate immortalized cells and we don't consider them, the DNA is the same, right? So yes, the sex of the cell is the same, but how they respond to the environment is going to depend on what kind of steroid hormones they have in their milieu and whether or not they express steroid hormone receptors, right? So you can get different responses even within the same sex, depending upon other factors, like whether the cells are endowed with receptors and what they're floating in. Okay, so what I think we need to do is making lemonade out of lemons, right? And it's trying to persuade you that the world isn't as grim as you think it is. Okay, so cells show, this is an example which was being used in this paper as sort of a drawback of why should we study sex differences or try to include cells from two sexes. Cells show sex differences in vivo, but not in vitro when cultured under identical conditions. Okay, so darn, we see it when the cells are in the body, but we pull it out. And so, okay, so, that's useless. But I think it actually tells you something important. And it's probably that the sex chromosomes aren't actually behind the sex difference, right? If it goes away, when you take the cells out of their native environment and grow them in a dish, well, I think that's telling you something else is behind that sex difference. And so what is that something else? I think there's a good bet that the sex differences in gonadal hormone levels at the time of cell harvest are involved in the sex difference seen in vivo. All right, so that's a very likely outcome when you are dealing with this sort of, when you're dealing with this outcome, this is a very likely explanation. Another possibility is that you're seeing a sex difference in vivo and it stays when you take it out and put it in culture, right? Now you have the question of, is it the sex chromosomes or is it something about the hormone environment early on that program those cells to be different in their adult state? And that's something that Marc will talk more in detail about. Okay, another example is there's no sex difference in the normal system, but when perturbed for example, disabling a key signaling molecule, all of a sudden a sex difference emerges, right? And that's, wow, that sounds confusing. But again, I'd like to argue that there's value in that. Depending on the nature of the effect, this could mean that it's nature's way of basically evening the score, right, and making things the same. And there's considerable precedent in terms of, given that there's differences in hormones, that sometimes sex differences in the way the nervous system is organized, I happen to be a neuroscientist, is actually to achieve the same result in males and females. But another possibility that I find more interesting is this idea that when you disable a key signaling molecule and a sex difference emerges, and and something happens, it can begin to help us understand sex differences in the prevalence of certain diseases. And so, for example, this has happened to a colleague of mine where disabling a particular signal molecule in the enteric nervous system reveals a sex difference, and all of a sudden he has an inroad for understanding perhaps why irritable bowel syndrome is more common in women. Okay, so just to wrap up, as they did in the paper, here are their recommended steps. Develop your knowledge base around sex and gender. I think that's a good idea. And you can read the literature, but I also encourage you to team up and make friends with the experts on campus. And there's a bunch of us here. And we like to talk about sex, right? That's our favorite topic. Address sex-gender issues when reporting and interpreting findings. Yes, that's a pretty obvious one. Here's one that I don't agree with. Introduce a small pilot intervention and report what you find. I believe that this is a dangerous strategy. I do not recommend it, but there is some controversy about whether this is actually a strategy that NIH has blessed. And Laura perhaps will comment more on this. My own perspective is that if you do a small pilot experiment and you don't see a difference because you have such severely compromised power, you don't know anything. And that to me is just as unfair as studying only males, right? I mean, what have you really learned? And so I don't really see the merit of that recommendation. And then finally, just before I turn this over and hopefully get a little bit more input from Laura on the third point, use your influence to raise the profile of sex-gender issues. Well, I would say perhaps that's the job of Marc and myself and other experts in the field, and that it's really not your job at this point if you're trying to navigate this whole sex-gender landscape for the first time. And so that's all I have to say. And Laura, did you want to share a little bit in regard to your perspective of pilot and what the study sections are saying?
Okay. So I think that there's been discussion, so I've been on committees. Well, first, I'm on a study section, and I've noticed that there's been increasing grants addressing these. So in February, in the spring, it was barely addressed, sex as a relevant biological variable. And then this October, we had grants that were addressing them. And as a study section, we were rigorously reviewing that. But in the case of my study section, we were looking just to be sure that it's addressed in some way, that there's an acknowledgement of sex similarly to the and providing a rationale similar to the rationale for the age of the mice, predominantly the age, because we randomly pick an age and, we pick a sex and we set up our experimental design with these things and the length of our treatment and all of these things. So in my study section, we looked at making sure that the grant at least acknowledged the difference between male and female in their design. So, but I've also been on committees where there's been a debate about, because as researchers, we want a rigorous study. And so to do a rigorous design, if you're going to do both males and females, then like you point out, Cindy, that you need you need many, many, many more animals. On the other hand, when we pick an age, for example, an age, so we'll like pick another variable, but you only need, some people put in an N of 5 in their proposal to suggest the importance of a difference between two age groups. And so that's preliminary. It's not rigorous yet, but it's pilot data suggesting a difference. Similarly, they could put it that there is no difference, and we as a study section would probably say, okay, there is no difference, even though the data provided isn't fully powered, because that's all that we ever judge the grants on. We don't judge them as full published papers because we're looking to see if there's a hint that something will work out. So those are things that go through my mind about, does this mean that and for many scientists, I feel that there's, to do the full rigorous analysis of male versus female differences is its own grant on its own, and it's additional to some of the original questions studied. So I find it a very complicated issue. I see both sides, I guess. So is that what you wanted to hear? Okay.
Yeah.
We can talk more about that.
And I've been talking to some other folks too about this. And so I just want to, so Cindy talked mostly about the FASEB paper. I'm mostly going to talk about the J Neuroscience paper, which, as she says, is very easy to understand, very practical, and I think really does a good job. I just want to start off by saying that I think probably the main reason all the research has focused on men is probably not really well justified. I think the concern always is there's more variability in females than in males, with the idea of the ovulatory cycle that somehow gets into that. But you know, that's a perfectly feasible hypothesis, but it's not clear to me that the data support it. And in fact, I think for the biomedical research up to this date, I think that's an idea that's been out there that nobody's been looking at. And you know, it's not much different from attributing all female problems to their ovaries, right? Which in our Western culture is a long history of that. And what I thought was really interesting was how when Prendergast et al., which is one of the other studies that's in your list, when they did a meta-analysis where they looked for all the studies where both sexes were used and were both reported, and looked at how much variability there was in measures, including behavioral measures, which probably have the most variability. In fact, it wasn't statistically significant, but in fact, there was less variability in females than there was in males. How about that? And the other point that they made was, in fact, the biggest variability is whether animals are housed singly or in social groups. And especially for males, that makes a tremendous difference, because in most rodent species, when males are isolated by themselves, they're going to become much more aggressive. They're a completely different sort of individual than if they're socially housed. And if you start looking around, you'll be shocked how many publications out there where they don't mention whether the males were housed socially or individually. It's not thought to be, so they're avoiding females and then ignoring this much greater source of variance. Anyway, I just want to make that point that I really think that I really do think it was a matter of bias and not, why did it take till 2014 for somebody to actually ask the question empirically? So why should you look for sex differences? I just want to echo Cindy's theme that that you should, and that in fact it's good for you, and maybe not even in a way that spinach is supposed to be good for you. The problem is that it's still not clear what reviewers are going to do. So I was so glad Laura could be here, who's dealing with this now, and I've communicated with a few other people who've been on study section. So the first thing I want to, all this about grantsmanship, as Cindy says, the first thing is if you do not address this, that's almost certainly fatal. You must address this, even if it's to say, I'm studying prostate cancer, and so of course we can't do it in female. I would, even in that case, I would make room for it somewhere to address it. And for virtually everything else, it's going to be an issue. I mean, as Cindy says, once you start being aware of sex differences, it's difficult to not find them. And I'll say in the neurosciences, I would say everyone assumed that there had to be sex differences in the brain because, of course, there's sex differences in behavior on these animals we study. But nobody looked for any sex difference because everyone assumed that they'd be really hard to see. And then once they started looking for sex differences, it turned out it didn't matter which species you looked at or what part of the brain you looked at. If you looked carefully enough, it turned out there was an average difference between males and females. They were just everywhere. And I hope this doesn't discourage you, but I think that whatever it is you're studying, there's likely to be sex differences. But this isn't a bad thing. This is a great opportunity for you. So again, in terms of grantsmanship, I would make sure there's a separate paragraph with that heading, sex is a variable, and you talk about this there, because if the reviewers, it's not enough sometimes to address the issue. If the reviewers don't happen to see it, they'll ding you for it, and then they get to the meeting, and another reviewer says, "Oh, they did talk about that somewhere," but by then, you're already done. I mean, because they came to that meeting with an idea about your score, and one of the ways that study sections don't work is once people have that anchor point, what they think about, it's very difficult to get them. Those of us that have been on study section, you would be amazed. Someone will say, "Well, I'll give this a, X, because X, Y, because A, B, C." And then the other members will say, "Oh, but A's here and B's there." Oh, well, okay. And then they'll move X by 0.1 or something. I mean, people have these. It's just one of our cognitive biases. Once you're anchored somewhere, it's very difficult for us to move away from there. So you want to make sure they know exactly where you address this, and go at it from there. So you found sex differences. No, don't. You're going to be fine. In fact, this is a reason to celebrate, because this offers an opportunity for you. Science is, the whole procedure of science is looking for differences, right? That's what we do all the time. We're looking for, we did this manipulation, and did it change things? We compared these cell lines, and are they different, et cetera. And so to the extent that sex provides, that there is a difference, this gives you another opportunity to manipulate the system and see what's going on. And I just want to say, and I'm going to talk some more about this, because this paper is also a great practical source of what to do once you find sex difference. But I just want to get back to this issue that Laura brought up, which is, it really is true. If you read and I've heard this from other people too, if you read the guidelines, and I feel like I even understand how this happens. NIH says you don't have to have twice as many animals. You can do some pilot work, and see if there's a difference or not, and then you can address that. Now, I'm pretty sure the reason they said that is they said, "Oh my gosh, if we tell everybody they got to do both sexes, we're going to have to give them twice as much money, and we don't have it." And so somebody said, "Oh, well, we'll tell them to do pilot studies. And that way it won't cost that much more." I mean, sort of whistling past the graveyard. So that's, and the NIH programs are instructing, I don't know if this is true in your case, they were instructing the reviewers to accept pilot data, right? To accept that approach. And if there isn't much, then it's, then they don't have to address that sex. But the whole job of reviewers is to find something wrong with your grant, right? They have to, because they don't get to fund nearly enough. And and as soon as you have something underpowered, as Cindy says, you've made yourself vulnerable. And so I think, I don't think the reviewers are going to be able to follow those instructions. We are so primed to look for problems that if it's underpowered, I think that's going to hurt you. And so I would say use that very sparingly, and try to avoid it if you possibly can. That's my advice. And so what happens if you do find a sex difference? I just want to say that the McCarthy et al. Paper is really very practical, and it goes step by step, and they even have these flow diagrams. I'm just going to show you briefly to what to. So the first question is, if you find a sex difference. So all of this is if you find a sex difference. The first question is, well, there are typically sex differences in circulating hormone levels. Are those responsible for it, right? Could well be. And most of the sex differences in hormone levels are differences in gonadal hormones, and the others are mostly due to the difference in gonadal hormones. And so if the first thing you do if you find a sex difference is you follow this research program A. They don't call it that, but I'm going to show it to you in just a moment. And if you manipulate hormones in adulthood, and you don't get rid of the sex difference, it's still there, then you go to research program B, and which is, as Cindy alluded to, in mammals at least, most sex differences that are, most sex differences are due to either exposure to androgens in adulthood or exposure to androgens early in life. You know, the sex differences in physical structure among mammals are mostly due to hormones from the testes, androgens like testosterone, masculinizing the periphery in males, and in the absence of testosterone early in life, the periphery will be take on a feminine phenotype. And so, if adult sex differences in hormone don't seem to explain the difference, the most likely other mechanism to explain it is due to sex differences early in life. We'll talk about that. And then, of course, occasionally you have to manipulate both. Not so good, but we'll talk about that. And very rarely, there are a few examples. Very rarely, it might be that hormones have nothing to do with the sex difference. Not usual in mammals. But if so, then there are things you can do to look to see whether sex chromosomes are directly involved in this. And I would say that although, I mean, given the vast number of sex differences we know in mammals so far, these sex differences due to sex chromosome, they're clearly the vast minority. But on the other hand, you know, if you see one of those, it'd be very exciting because it would indicate that you really have a handle on something about what is it about having a Y chromosome or having two X chromosomes that causes that. So just I don't want to go through the whole paper, but just to give you an idea. So here's the first strategy, that program A, part 1, is, you know, do adult hormones responsible for this? And in our field, Cindy and I, the joke is, you know, the joke about how to cook chicken dinner is step 1, steal one of your neighbor's chickens. For us, how do you study sex difference? Step 1, take out some gonads. And so the first thing you do is remove the gonads in males and females so that you're going to get rid of the source of sex differences in hormones, and then see whether that gets rid of the difference. So you gonadectomize them and if equalize hormones, and if there's no difference anymore, well, okay, you conclude the sex difference is hormonally modulated in adulthood. If you do still see a difference, then there's some other possibilities. The other thing is to deprive hormones in both cases, or to compare males to males, et cetera. So I won't belabor those various possibilities, but they go through it step by step to tell you what you could include and what you would do next. And the other thing I want to point out, here at MSU, we are so lucky that if you haven't done these surgeries before, we have CAR that's happy to come to your lab and show you how to remove testes and how to remove ovaries and things like that. Show you how to do it, you know, the right way and safely and efficiently. And so that's one thing that can happen with Part A. Then also, if you're looking at for differences due to hormones circulating, there's the next bugaboo, Part B, is will the ovulatory cycle be important for this? If you have evidence that the ovaries might be important for the sex difference, then the next question is it due to high estrogen levels or high progesterone levels or changes in progesterone levels, et cetera? And this article, they recommend, and I think this is really great advice, you might start thinking the first thing you do, oh my gosh, first we had NIH requiring me to have twice as many animals for sex differences. Now, for my females, I've got to look at 4 different stages of the ovulatory cycle. Now I've got 5 times as many animals that I've got to pay my per diems for and try to find a way to work with. And that would be a terrible idea. Almost always that's a bad idea. And in fact, you don't need to do that, as they point out. The first question is, well, just look to see if the sex difference is there even when you don't control for the ovulatory cycle. And so then they have another chart where they talk about this. So this is, you measure adult levels. There's several things you can do. First of all, just look, and if the mean is different, but the variability is equal, right? If you know your females are in 4 different stages of the ovulatory cycle, and yet they don't show any more variability than the males, then that's a pretty good argument to the study section as to why you don't need to go rushing into looking at comparing across the ovulatory cycles. Although again, in terms of grantsmanship, you explain that that's why you don't have to do it now, but you indicate that in the future that is one of the things that you'll address, right? So that they don't then ding you for not addressing it. And if there is more variability if the variability is greater in males and the mean is different, then there are other things that you can assess. And then program B that they talk about. Oops, sorry. Yeah. Well, then the other program they talk about is, okay, we equalize hormones in adulthood and we still have a sex difference. How can that be? They've got the same hormones and yet they're showing differences in your in measure. And for mammals, most of the time, what's going on is the difference is due to the fact that the males were exposed to testosterone early in life and the females were not, or not as much. And so then they have this great chart of what you do about that. What sort of things you look at. You either you give the females the male hormones, or you treat males with antagonists, or you remove the testes in males, and you look to see what happens. And here's this, this decision tree that they offer you. And then another possibility is it could be that you do both those things, and it turns out you have to manipulate hormones early in life, and you have to make instill the difference in sex difference in hormones later in life. And so, nobody likes to have more work. But on the other hand, I'll just say that if you find out, whatever you're looking, if you find out that testosterone early in life changes something about it, and that testosterone late in life changes it, if either one of those happens, suddenly you've got a new way to manipulate the system, right? Now you manipulate it. And the cool thing about hormones is you just call up Sigma Chemicals in St. Louis and they send them some to you, right? And so you can make this manipulation relatively easily. And then the last thing, and this is the most unlikely outcome, there are some sex differences where it doesn't seem to matter about hormones any time in life. It's something that the X chromosomes and the Y chromosomes do differently. And there aren't many examples of that, but you might worrying about it, what would you do if you had that? And again, it's a great how-to. They explain to you that there are things you can do, because you can get mice that have 2 X chromosomes, but also have, but don't, let's see. You can get mice that have an X chromosome and a Y chromosome, and yet they still have ovaries, because you can get them where they've disabled the one gene on the Y chromosome that causes the gonad to develop as testes rather than ovaries. And likewise, you can get animals that have XX chromosomes, but also have testes, because they've got an extra gene going along that will cause the gonad. And so you can compare, they call them the 4 core genotypes: XX females, XX males, XY females, XY males. And they tell you how you can compare those animals and look for an effect of sex chromosomes directly. And so I just want to emphasize again that all of these are turning an obligation into an opportunity. I think once you start looking for sex differences, I guess I'm not willing to bet my own money on it, but my guess is that you're likely to see sex differences, and that I think that then that gives you another way to look at your system. To try to put it another way, if you find out the most likely outcome is testosterone at some time of life is causing this difference, well, now all you have to do is find out where's the testosterone going, what's it doing there, how is that affecting your measure, et cetera. I mean, suddenly you've got an entryway into in the problem that you might not have expected otherwise. And so I think that's all I have to say. Thank you.
Well, thanks to all of our speakers. We're going to have questions in a minute. So So let's open the floor to questions. While you're thinking, let me ask Marc and Cindy a question first. That was a great presentation in terms of strategies, but where do people go to know how much hormone to give back? You know, so you do the ovarectomy or the gonadectomy, and you know, you don't want to get into a toxic situation with overdosing estrogen or testosterone.
Yeah, it's a great question, and again, Cindy answered it on this campus at least. We've got plenty of people on campus that study these animals. So yes, for example, estrogen can be quite toxic, right? And so the people on this campus. Carr can tell you how to do the gonadectomies, and there are lots of people on campus that can tell you relatively easy ways to get to come close to recreating physiologically normal levels of these hormones.
Thank you. Because I think that could be an important factor. Other questions? And please come to the microphone with your questions, so that, since this is being recorded, so that you can all hear this again later.
My question is, though I'm not an animal researcher, so it looks like that cost of doing, having female rats or mice is the one big huge factor in doing sex differences in biological research, and that seems so unfair, actually. As a female, I feel, really, with health disparities and all that between males and females, if this is the only thing, because it's so much more difficult and your sample has to be so big, and cost is the only consideration, really, I think it's really unfair to more than half the population. So,
to not do it.
yeah, not do it, actually.
Exactly. You know, I think, like Marc said, how did we get to 2014 and ask the question? So
Right.
it's
Yeah, in NIH's
inherently
mission.
obvious when you think about it. Go ahead.
No, the NIH mission isn't to improve the health and welfare of mankind, right? It's to improve the health and welfare of humanity, yeah. So I agree, and of course, I'd love for the NIH to give us twice as much money to handle twice as many animals, but I think, it's
Well,
just realistically, we're in the climate
To be fair,
we're in.
I think NIH would love to give you twice as much money. Congress has to give it to them.
Touche.
So we'll do our shtick for advocacy here. Write Congress if you think more money needs to go into research.
I have an additional comment, and that is the animal number issue and the idea of pilot. I mean, piloting is going to be okay, maybe okay, if you find a difference, right? But even there, what we're hearing, this flies in the face of the expectations for high rigor, right? If you're being, if on the one hand they're really dealing in a serious way with rigor and reproducibility, but then suggesting that we can make conclusions that we don't need to include two sexes because we did this small pilot and we didn't see a difference, right? I mean, I understand, we all use pilot data when we're submitting a grant. In fact, we may have fully powered studies and we're still going to call it pilot data because otherwise NIH won't give us the money, right? But you see that there is a contradiction there in the underpowered studies. You can acknowledge that it's pilot data, but you still have the challenge of dealing with what is your specific aim, what is the experiment going to be, right? Pilot data is the foundation on which you build your grant. I just don't know how you can be sanctioned for doing underpowered work when at the same time you're supposed to do well-powered work to achieve the rigor and reproducibility issue that is also being discussed at NIH.
So I would say your logic is sound. But the meeting, one of the meetings that Laura referred to was a national meeting at the American Physiological Society Science Animal Care and Experimentation Committee that we both happened to attend. And representatives from NIH were there, one from the Office of Women's Health. And I mean, we had a robust discussion about this. I mean, this discussion went on for about an hour and a half, pointing out all the pieces, wouldn't you say? And his final word was that, you know, the pilot data, if there's an effect there, should be robust. You should be able to see it. But they were willing to miss something to have people start thinking about this and realizing that science is incremental. Science doesn't take giant steps. And if they can get people to start thinking about these issues in their experimental design and their planning, then progress is being made. So, you know, it's a big step to start thinking about sex differences in the context of whatever other system you're studying. And I think that's part of their rationale. Of course, I think Marc nailed it too. He said they just don't have enough money to do it. Laura, do you have anything to add to that? Did I capture that discussion?
I mean, what I really liked was the analogy to age. So, you know, for bone, there are different bone responses in young mice versus adult versus more older. So the whole remodeling is quite different. And so there are age differences. So, you know, that seems important too. You know, so you have to make some choices, and so that's where I think for the piloting is more about this is sort of guiding my boat down a certain way. Here's the well-powered data showing that there's an effect in this. You know, in my case, we start with females predominantly, and then we do the ovaryectomy. I mean, that's what we do because of bone loss with menopause. And then we've been going into males to see, oh my goodness, they're quite different. But, you know, I just wonder if every grant, so, you know, I'm just over here pondering this, that if we all are addressing the biological sex as a major variable in doing males and females, that takes away from the depth of our scientific study, and in which case, and then I'm wondering, will reviewers start seeing this over and over and think it's not innovative in some way? You know, and I hate to, you know, but these are the crazy things going through my mind. And then the same with the, you know, I mean, you bring up a really good point. So reviewers will say this isn't rigorous data, you know, and I don't know. I don't know what the perfect thing is. But I think that being on a reviewer study section, it's a way that reviewers could control other reviewers too. So if somebody sees, let's say they see a sex difference and but it's not fully powered, and they're using it as a rationale to study it, other reviewers could say, well, there's a trend here, it seems important to follow up. You know, so I think we can help as reviewers.
In terms of grantsmanship, it seems that the strategy you presented would be very important, you know, in terms of being able to cite that paper or others like it to say, you know, with the pilot data that I've collected so far, I haven't seen, we're going to keep looking, and if we find something, we'll be employing the strategy of McCarthy et al., that kind of thing. You know, there are tricks to the grants personship here, and that may be a way through it initially. You know, we're still waiting for it all to fall out as to how the study sections are going to integrate this over. How many study sections are there at DRG? Hundreds, right? So it'd be interesting. Questions from you? Any? Okay.
I want to make one additional comment, which is more a comment of suspicion, but I think you've kind of alluded to it as well. Is, you know, so this is what study section is being told this year, because it's definitely on a ramp, right? And so what study sections are told this year is probably going to be different than what study sections are told next year. And it is this business of trying to, get everybody on board. This, so in a way I sort of compare it to, that all our NIH supported research papers had to be publicly available, freely available, right? And now, we can't write a progress report, and we certainly can't get a competitive renewal funded without making sure that our NIH supported research publications are available, are publicly available, right? But it was several years between NIH having this mandate and then really enforcing it. It took several years to have that happen. And so I think we're probably going to see the same trajectory here, where this is one level of seriousness, and it's gonna ramp up. And I think one of the expectations has to be that perhaps it's fewer aims, that if you can't get more money, but they want more animals because you're going to be looking at both sexes, then you're going to have to do fewer experiments. You're going to have to, if it's the same amount of money. And so that's another adjustment, and I don't know if that is getting discussed at the study section level, as to whether the reality is, it's, you have X number of dollars and you can't take on as many aims.
The playing field keeps changing. And we have to remember this is one of the aspects of rigor and reproducibility. It's not the only aspect. That definition of the research conditions, I think, will become more and more important. Whether it's cell lines, whether it's reagents, whether it's animals, whether it's the experimental design and making sure that is laid out clearly, and publication. I mean, there are lots of pieces to this. So, any other questions? Well, thank you for attending today, and the conversation will continue. We have a date set up, I think, in the spring for another one of these, but keep an eye on your email, and we'll see what I think we're trying to aim towards experimental design for that, for the topic of that session. But we're still planning. So thank you all for coming, and happy Thanksgiving.